Hello my dear students, welcome you all. Today I am going to teach you the very interesting topic monoclonal antibodies. Monoclonal antibodies is used as targeting carrier system or site-specific drug delivery system. So let us now discuss in depth. To understand the concept of monoclonal antibodies, we will have to first understand the concept of drug targeting. So, what contents would I cover in this topic? The introduction about drug targeting, the introduction about monoclonal antibodies, the role and need of antibodies in immune system, a structure of monoclonal antibody, its types and production, drug conjugates along with monoclonal antibodies for drug targeting, applications of monoclonal antibodies, advantages and limitations, and FDA approved monoclonal antibodies in the market. So, we will now discuss about drug targeting. Drug targeting. Targeted drug delivery system is a special form of drug delivery system where the medicament is selectively targeted or delivered only to its site of action and not to the non-target organs or tissues or cells. It is a method of delivering medication to a patient in a manner that increases the concentration of the medication in some parts of the body relative to others. Targeted drug delivery seeks to concentrate the medication in the tissues of interest while reducing the relative concentration of the medicament in the remaining tissues. This improves efficacy and reduces side effects. What are the reasons for drug targeting? There are some potential reasons for drug targeting are drug instability, low absorption, short half-life, large volume of distribution, low specificity, and low therapeutic index. These all are the potential reasons in conventional drug delivery systems. That's why nowadays we choose drug targeting carrier to target the cells or tissues by delivering the drug through a carrier system or targeted carrier system. There are various drug targeting strategies which are passive targeting, inverse targeting, active targeting, ligand mediated targeting, physical targeting, dual targeting, double targeting, and combination targeting. Let us now discuss in detail about monoclonal antibodies. These are also known as drug targeting particulate carrier system. Antibodies are produced by a specialized group of cells called B lymphocytes. We all know about immune system in brief. When a foreign antigen enters the body due to immune response, B lymphocytes develops into plasma cells and liberates antibodies or immunoglobulins of various types like IgA, IgD, IgE, IgG, and IgM. These are various types of immunoglobulins or antibodies. This is the structure of immunoglobulin. here these are the antigen binding sites. When an antigen enters into our body, our immune system or due to immune response, B lymphocytes are produced that liberates antibodies or immunoglobulins. So antigen and antibody complex are formed and the antigens are killed. What are antibodies basically? An antibody is a protein used by the immune system to identify and neutralize foreign objects like bacteria and viruses. Each antibody recognizes a specific antigen unique to its target. Monoclonal antibodies are antibodies that are identical because they are produced by one type of immune cell, all clones of a single parent cell. What are polyclonal antibodies? These are antibodies that are derived from different cell lines. They differ in amino acid sequence. This is the structure of antibodies. In the upper end, these are antigen binding sites. This is light chain and this is heavy chain in both the strands or in both the structure. Role of antibody in immune system. Each antigen has specific antigen determinants, that is also known as epitopes, located on it. The antibodies have complementary determining regions. These are mainly responsible for the antibody specificity. Each antigen has several different epitopes on it. They are recognized by many different antibodies. All these antibodies thus produced act on the same antigen. Hence, these are designated as polyclonal antibodies. Need to develop antibodies. In general, naturally produced antibodies are non-specific and heterogeneous in nature. Hence, there are several limitations in the use of polyclonal antibodies for therapeutic and diagnostic purposes. Thus, there is a need for producing monoclonal antibodies for different antigens. What are the advantages and limitations of monoclonal antibodies? If we talk about advantages, they are homogeneous in nature. They are specific to a particular antigen with a particular epitope. For example, rituximab, it is a anti-CD20 product, is a good example. This antibody is used for the treatment of lymphoma. And there are some of the limitations of using monoclonal antibodies are, as they are specific to a particular antigen, they cannot distinguish molecule as a whole. Sometimes they cannot distinguish groups of different molecules. For example, presence of retroviruses as a part of mammalian chromosomes is not distinguished. The presence of some of these viruses is detected in hybridomas. This poses a great danger since there is no guarantee for monoclonal antibodies produced is totally virus-free. Let us now discuss about types of monoclonal antibodies. There are mainly 4 types of monoclonal antibodies. These are murine, chimeric, humanized, and human. Let us now discuss about production of monoclonal antibodies. Monoclonal antibodies are produced by hybridoma technology. First, we introduce an antigen to a mouse. Then we extract the spleen cells of that mouse, fuse them with myeloma cells. Myeloma cells are tumor cells. Myeloma cells are basically tumor cells. So when we fuse tumor cells with the spleen cells of the mouse and fuse them together to get a hybrid cells, or that is also called as hybridoma, then these hybrid cells, when we introduced in the het medium or when we culture in het medium, then we select for positive cells. Then finally, we will now harvest monoclonal antibodies for drug targeting. There are various steps involved in the production of monoclonal antibodies. There are basically 5 steps involved in the production of monoclonal antibodies are immunization, cell fusion, selection of hybridoma, screening, cloning and propagation, characterization and storage. Immunization. Immunize an animal, for example mouse, by injecting with an appropriate antigen along with adjuvant. Then we inject the antigens at multiple sites, are repeated several times for increased stimulation of antibodies. 3 days prior to killing of animal, a final dose is given intravenously. Spleen is aseptically removed and disrupted to release the cells. By density gradient centrifugation, lymphocytes are separated from rest of the cells. The second step is cell fusion. Lymphocytes are mixed with myeloma cells or tumor cells and is exposed to polyethylene glycol for a short period. The mixture is then washed and kept in a fresh medium. The mixture contains hybridomas, free myeloma cells and free lymphocytes. The third step is the selection of hybridoma. The above mixture is cultured in het medium or HET medium for 7 to 10 days. Due to lack of HGPRT enzyme in myeloma cells, salvage pathway is not operative, hence free myeloma cells are dead. As the lymphocytes are short-lived, they also slowly disappear. Only the hybridomas that receives HGPRT from lymphocytes are survived. Thus, hybridomas are selected by using het medium. Suspension is diluted so that each aliquot contains 1 cell each. These are cultured in regular culture medium, produced desired antibody. The fourth step is screening, cloning and propagation. Screening is done for antibody specificity. Common tests like ELISA and RIA are used for this. ELISA is enzyme-linked immunosorbent assay and RIA means radioimmunoassay or radial immunoassay. The single hybrid cell producing the desired antibody are isolated and cloned. Usually two techniques are commonly employed for this. First one is limiting dilution method. In this, suspension of hybridoma cells is serially diluted, so the aliquot of each dilution is having one hybrid cell. This ensures that the antibody produced is monoclonal. Another method is soft agar method. In this method, the hybridoma cells are grown in soft agar. These form colonies and the colonies are monoclonal in nature. The fifth or last step in the production of monoclonal antibody is characterization and storage. Biochemical and biophysical characterization are made for desired specificity. It is important to note the monoclonal antibody is specific for which antigen. Monoclonal antibodies must be characterized for their ability to withstand freezing and thawing. For drug targeting, we prepare drug conjugates. Here are some of the examples of drug conjugates. First one is toxin conjugates. This is also known as immunotoxins. For example, diphtheria toxin, ricin have been conjugated to the tumor-specific antibodies. A chain is cytotoxic and B chain is non-specific. Hence, B chain is removed and the toxin is conjugated to tumor-specific antibody. Thus, we increase the specificity of the toxins by using monoclonal antibodies as active drug targeting systems. Another example is drug immunoconjugates. Agents like chlorambucil, an anti-cancer drug, methotrexate, it is also an anti-cancer drug, and doxorubicin. These all are anti-cancer drugs. These are conjugated with tumor-specific antibodies. For example, doxorubicin BR96 immunoconjugate for Lewis antigen found on the surface of tumor cells. These are some of the applications of monoclonal antibodies. Diagnostic application, biosensors and microarrays. We can use monoclonal antibodies as biosensors and microarrays. Therapeutic applications, monoclonal antibodies are used for transplant rejection or in the case of transplant rejection, in the treatment of cardiovascular diseases, in the treatment of cancer, infectious disease and inflammatory diseases. Clinical applications, purification of drugs and imaging the target. And future applications of monoclonal antibodies would be fight against bioterrorism. Bioterrorism, what does it mean? Bioterrorism is, we can say, is like a biological weapon. It will be used in future. It is expected that there will be some countries which will use this type of bioterrorism. So it will be very useful in that case also. Monoclonal antibodies would be more helpful in that case also if it happens in future. There are some of the monoclonal antibodies which have been approved by FDA. The example for those antibodies are trastuzumab, bevacizumab, cetuximab, panitumumab. These are used for breast cancer, lung cancer, colorectal carcinoma respectively, and the target sites for these antibodies are the HER2, VEGF, EGFR and EGFR. These are the target sites for these antibodies. So thank you all for watching this video. If you like this video, please hit the like button, subscribe to our channel and press the bell icon for getting instant notification when I upload another video of your interest. Thank you once again.